First of all, I want to express my gratitude to the organizing committee for allowing me to stay here in this symposium and in my city, Valencia. I hope you enjoy and visit this beautiful city. Today I'm going to talk about the anticarcinogenic effect of phytosterols and or β-cryptoxanthin in colon cancer cells. You know this sentence, let food be your medicine and medicine be your food. This was the famous sentence pronounced already 25 centuries ago by Hippocrates, indicating the importance and conditioning effect of food on health. Nowadays, with modernization and the related lifestyle behaviors have made possible the appearance of a chronic disease era, and also it is well known that a balanced diet with a regular intake of fruits and vegetables together with a healthy lifestyle are essential factors to diminish the incidence of different chronic degenerative diseases, including different types of cancer and particularly those of the digestive tract. But this is not new. Because in 1974 it was published by the World Health Organization a report led by the Canadian Ministry of Health, McLellan, stressing the different determinants on health, illness and death, and this report indicated that lifestyle practices have an account in determinants of health of more than 50%. Including toxic habits, diet, exercise, ways of relating, and so on. This report supposed the beginning or the improvement of health promotion, and is important for people to manage their own behavior in order to improve their own health. Like individuals, societies have a life cycle, and for example, as we can see here in the slide, in low-income countries, there is prevalence of infectious diseases in blue color in combination with nutritional deficiencies. When the society grows up, there is a shift to a non-communicable diseases, particularly in urban populations, and when this is the trend observed in high-income countries, and it is projected to prevail in all over the world in the future. We have seen that the chronic diseases are a leading cause of deaths all over the world. And for example, data from 2002 indicated that the leading cause of death were cardiovascular diseases with almost 17 million of deaths, followed by cancer, 7 million, then lung chronic diseases, 4 million, and finally diabetes mellitus with 1 million approximately. In the particular case of cancer, colorectal cancer, we can see that there is a worldwide international variation in the incidence of this disease, particularly higher in developed or westernized countries, and according to the so-called nutritional transition, in low-income countries there is a change from infectious diseases to an increase on chronic diseases. This type of cancer is the third most common in men and the fourth most common in women. In the particular case of Spain, of my country, males are situated in the 9th position of incidence and women in the 21st position, indicating or stressing the prevalence of this disease in our environment. This disease has different etiology. On the one hand, the heredity factor, and on the other hand, is considered an environmental disease affected by culture, social status, and different lifestyle practice. Different epidemiological and animal studies have linked the unhealthful diets with low fever, saturated fat, with the origin of colorectal cancer. Typical risk factors include toxic abuse, alcohol, smoking, unhealthful diet, and also important, physical inactivity. The evolution of this disease needs at least two minimal factors to promote the progression. One is the deregulated cell proliferation, and the other is to escape or avoid the programmed cell death, formerly called the apoptosis. The evolution of this disease presents a transition from a normal epithelium, different states of adenoma, and a final carcinoma state. Then it can be disseminated or spread to other organs in the body. But on the other hand of the balance, we have chemopreventive agents like phytochemicals found in the diet, and they can act in the progression of this disease by two modes. On the one hand, blocking or inhibiting the proliferation of this disease. The blocking agents normally try to avoid the damage to DNA, and the suppressive agents try to slow down the unregulated progression of this disease and also to avoid the new blood vessels formation, the angiogenesis. Okay, once situated, the importance and the prevalence of chronic diseases, and in the specific case of colorectal cancer, we're going to talk about the functional foods. All of you know that these foods are spectacularly increased in last years and continue with the development. There are a wide array of food ingredients, functional food ingredients in the market, and according to the industry forecast of this year, the 12K trends in 2014, nutritionally functional foods are going to be the leader group for the next 12 months. Functional foods are typically classified in three different groups. On the one hand, the classical group containing vitamins, minerals that have been used since several decades ago. There is also a group of different fiber and carbohydrates, including beta-glucans, sinulin, so on. And there is a third group comprising bioactive compounds. We're going to focus today in this group, bioactive compounds, and especially in phytosterols, the main dietary phytosterols, β-sitosterol, campesterol, and stigmasterol, and also in the carotenoid group, the xanthophyll β-cryptoxanthin. But why these compounds, most of you are wondering? Because they are subject of research in a Spanish National Research Project, which is carried out by my research group, the Villanotes group here in the University of Valencia, in collaboration with the vitamin unit of the Hospital Puerta de Hierro in Madrid. And these compounds are well known. They have been reported to have, for example, β-cryptoxanthin beneficial effects in bone remodeling markers, and phytosterols they decrease cardiovascular risk due to their cholesterol lowering properties. In the particular case of phytosterols, due to the low absorption, they can reach the colon in high quantities. So we make a question, what about their combined action against colorectal cancer to have a complementary action to this bone remodeling marker or hypocholesterolemic effect? The mechanisms of anti-cancer, antiproliferative activity of phytosterols and carotenoids are well known and comprise different activities, among others. They can alter the regulation of cell cycle progression, apoptosis, antiproliferative activity, also induce the activation of antioxidant enzymes to decrease oxidative stress. Okay, this is well known, but the mechanisms in colorectal cancer are not fully elucidated. And so we can make advance in this case for these compounds in colorectal cancer. So taking into account this background, the objectives of this study were to first evaluate the antiproliferative activity of main dietary phytosterols and or β-cryptoxanthin alone or in combination against a colon cancer cell line, CACO2 cells, and on the other hand, to unravel the biochemical and molecular mechanisms involved in their possible antiproliferative activity. To this purpose, we used the colon cancer cells, CACO2 cells. These cells were treated 24 hours with standard compounds, these xanthophyll β-cryptoxanthin and plant esterols at concentrations compatible with human serum levels, as reported previously in our group and the group of Madrid in collaboration. Different spectrophotometry, flow cytometry, and Western blotting analysis were used to check the possible antiproliferative and anti-apoptotic mechanism of these phytochemicals. First, we evaluated the effect of these phytochemicals on cell viability, and we observed that all treatments evoke a decrease in cell viability from 21% to 44%, with the exception of β-cryptoxanthin and campesterol at their lowest concentrations. We also saw that there were no great variations with different treatments, standards alone or in mixtures. Also, we did not show additive or synergistic effects, rather, although not always statistically significant, in the case of mixtures with high concentrations, low antiproliferative activity. Regarding cell cycle distribution, all treatments provoke different patterns regarding to control. In this case, all treatments evoke a significant rise in the sub G1 phase of the cell cycle. This is a hallmark of apoptosis associated with DNA fragmentation, with the exception of beta-sitosterol that evoke an arrest in the G2M phase of the cell cycle, halting the progression of the deregulated proliferation. The highest effect in sub-G1 phase was observed in the plant-testerol mix, pointing again the fact that there is no additive and synergistic effects, but possibly a pronounced impact of the stigmasterol. Then we tried to see which were the different apoptotic factors involved in this mechanism of antiproliferative activity, and we evaluated the externalization of phosphatidylserine to the outer part of the membrane, the decrease in mitochondrial transmembrane potential, and also the cleavage of specific DNA repair proteins like poly-ADP ribose polymerase. Here we have a representative image of the apoptosis determined by flow cytometry, and all treatments evoke a 2 to 4-fold increase in early apoptosis in the right upper quadrant compared with control, without significant differences among them. Also, we also observed that stigmasterol and both mixtures have a double proportion of cells in late apoptosis compared with control. One of the features of apoptosis in early stages is the decrease in mitochondrial membrane potential, and we can see that all treatments decrease the mitochondrial membrane potential between 31 to 36%, with the exception of beta-sitosterol that evoke a decrease but not statistically significant. This different behavior together with the G2M phase arrest indicate for this compound maybe a different mechanism of action possible. As indicated before, we investigated the effect of these treatments in the specific proteins. This poly-ADP ribose polymerase is a DNA repair enzyme that is cleavage by activated caspase 3. This is one of the main executors of apoptosis. In the Western blotting, we see that all treatments evoke a cleavage of this protein with the formation of this cleaved product of 89 kilodaltons and the concomitant decrease of the full-length protein of 116 kilodalton. The highest effect was shown for beta-sitosterol and the lowest for β-cryptoxanthin and the mixture containing β-cryptoxanthin. Then we want to study the effect of the treatments on the redox cell state, and so we determine the accumulation of reactive oxygen species that has been associated with cell death in cancer cells. All treatments, we can see that evoked a 10-fold increase in the content of reactive oxygen species compared with control. Again, with the redox status, we'll evaluate the total thiols, which this determination include reduced glutathione, which is the main non-enzymatic antioxidant intracellular compound. And contrary to that expected, all treatments evoke a rise in these total thiols particularly higher with the plant-testerol mix. We hypothesize, and as the bibliography have said, that this can be an adaptive or response mechanism of the cell trying to avoid the oxidative stress. In addition, it is not shown, but with all treatments we saw also an increase in the protein synthesis, and partly this could explain this effect, but it's not enough to overcome the apoptosis effect that exerted by the phytochemicals. Another aspect involved in the early stages of apoptosis is the intracellular influx of calcium in cell that activates a cascade of signaling pathways involved with apoptosis. With flow cytometry, we determined that all treatments evoke an increase in cytosolic calcium, specifically higher with the plant-testerol mix, in this case nearly an additive effect, and it is not significant, but a trend to rise with campesterol. And finally, we also determine the pro-apoptotic bad protein, which when dephosphorylated is activated and inhibits anti-apoptotic proteins like BCL2, and this is due to the rise in intracellular calcium levels. Western blot images indicate that all treatments evoke the decrease of phosphorylation of this protein, so activates this pro-apoptotic protein. Highest effects were shown with β-sitosterol, campesterol, and plant-testerol mix, and the lowest with stigmasterol and the mixture containing β-cryptoxanthin. So, in summary, and to have a global picture of the action of these phytochemicals, the carotenoids and plant-testerols, in the antiproliferative activity, we have seen that they produce an apoptosis effect. This apoptosis was due to a rise in cell cycle in the sub-G1 phase, and also to the externalization of phosphatidylserine to the outer part of the membrane. As initial triggers, we observe the increase in reactive oxygen species and also the intracellular influx of calcium that can decrease the mitochondrial membrane potential, dephosphorylate pro-apoptotic bad protein, so activating this protein, and initiating the caspase cascade that finally make the cleavage of the PARP protein, which is a DNA repair polymerase. So the conclusions of this study were, on the one hand, that β-cryptoxanthin and our main dietary phytosterols, alone or in combination, reduce cell growth of colon cancer cells up to 44% due to apoptosis, possibly through the mitochondrial pathway. No clear dose response was observed, neither additive nor synergistic effect for mixtures, but in mixtures, they retain the same antiproliferative activity as individual compounds, indicating the absence of antagonistic effects. Also important that the effects were obtained with concentrations compatible with physiological serum levels in humans and with reported bioavailability of these phytochemicals after regular consumption of beverages containing a mix of these compounds. These results highlight the importance of conducting antiproliferative activity assays with whole foods or real matrix foods rather than individual compounds to see the interplay of different compounds. Like we are talking here, which is the recommendation for this, individual supplementation or a holistic view with a whole diet? This maybe it has more sense. So in our group, we are currently performing assays with functional beverages containing these compounds, subjected to in vitro gastrointestinal digestion, in vitro colonic fermentation, to see the persistence or not of these antiproliferative effects in colon cancer cells. Before to end, I want to acknowledge all the people in my lab here in Valencia for the kind day they work, and also and especially all the people in the laboratory of biochemistry at the University of Palermo, where I did a brief research stay and performed this study. So nothing else. Thank you for your attention.
Thanks a lot for the very interesting talk. Are there any questions? Please.
Yes, there is some people that have mutation in ABCG5 and ABCGA protein, so they have a higher level of phytosterols in plasma. Is there any evidence that these people has less of this type of cancer?
I don't know. I don't know if they can benefit of this, but I know that there are polymorphisms, and people can have beta-sitosterolemia and accumulation, and there is a contradiction if they are good or not. But recently in Europe, different associations of atherosclerosis have indicated that the regular intake of phytosterols in the allowed proportions that are from 1.05 to 3 grams a day are safe, and they have no problem with this accumulation. But I don't know in this kind of people if they can take a benefit of this or have different problems related with colon. I don't know.
Thank you.
But this is also interesting to check specific people with polymorphisms.
We know that the bioavailability of phytonutrient is too low, in some cases 1-3%. And in your study, you don't see the effect of those response because it wasn't clear. Perhaps because you used the physiological dose in your study, in vitro study, which that's why you don't see the dose response. Are you planning to use like higher doses to see if there is any effect or not?
We have chosen this concentration because it's the physiologic according to bioavailability in serum. And now we are trying to conduct this exercise with cells according to data of content in feces, because it's the content that can reach the colon. And but we have made calculation and more or less are similar, a little bit higher, but more or less these effects. We have not seen a dose-response effect, maybe because there is or there are interaction among these compounds when they are taken up by cells, or maybe because there is a threshold to activate signaling cascade, and it is enough with low concentration, and with higher there is no benefit. No. This is maybe the hypothesis.
Okay, time for one more last question. I have one question. You said you are studying now. You point that the main activation area could be related to mitochondria. Do you have some estimation about the potential target?
In mitochondria?
Yeah.
No, this is a global measurement of the mitochondrial membrane potential, but not a specific target in the mitochondria.
What if you had that information?
Sorry?
What would you do if you had that information?
Well, if I have the information, this make more value for these compounds in order to make functional foods that not only lower cholesterol or have benefits in bone health, but also maybe in other chronic diseases like colon cancer, and we can exploit maybe a particular aspect. In fact, we are also conducting assays with eryptosis, that is apoptosis in erythrocytes, and maybe they can have an action against the oxidative stress that evokes the eryptosis, complementary to these other actions. So all of this can be very important.
Okay, thanks a lot.
Thank you.